Okay, so good afternoon, everybody. My name is Pilar Blancafort, and I'm going to talk today about some of the works done in the lab here at the Perkins, at the Harry Perkins, on exploring the therapeutic potential of honeybee venom and melatonin in targeting aggressive forms of breast cancers. And first of all, I wanted to thank the organizer for giving me the chance to talk today. To give you a little bit first background for this audience on the tumor types that we're working in the lab. So the lab is working, has been working now for many years on breast cancer. And breast cancer is one of the most commonly diagnosed cancer in Australia. 1 out of 7 women are going to be diagnosed of breast cancer. The second most common cause of death in women, and whilst the survival, the 5-year survival is relatively high, you think about 76 to 91% in 2001, there are still subtypes of breast cancer which are difficult to treat, and those ones are the triple negative subtype shown here, about 10% of all the breast cancers, and then the HER2 enriched, which is about 5%. So those cancers are still very recalcitrant, very difficult to treat. Ovarian cancer, in another hand, is the type of tumor with very poor prognosis in women. Definitely one of the reasons is it's diagnosed late, and there is a lack of biomarkers for ovarian cancer. The 5-year survival is only 42%. And very high recurrence of 75%, which is the rate where the cancer comes back. And this is incredibly high. It affects women that are young, less than 40 years of age. So what my talk today is centered basically in these 2 subtypes, so triple negative in breast and in ovarian cancer, so we call high-grade ovarian cancer, which are tumors that are aggressive and poor prognosis. And what's important, consequently, is that there are a lack of targeted treatments for these cancers. So in breast cancer, as I mentioned, the vast majority, about 80% of the breast cancers express the hormone receptors. So we're going to abbreviate this as HR in this talk. And so that means that the treatments available for these breast cancers are effective, and they target the estrogen and the progesterone receptors. So there are anti-hormonal treatments. And that's why these 80%, or that majority of subtypes, majority of breast cancers are relatively benign. They are relatively more benign. The triple negative breast cancers are abbreviated here as TNBCs. However, they lack the expression of hormonal receptors and another cell receptor that's called HER2. And so that means that the targeted treatments that are available for these HRs and HER2 enriched tumors are not effective in the context of triple negative because they don't express the targeted molecules. One thing I want to stress in this talk, though, that there are molecules on the surface that I'm going to go more in detail, and it's important to explain this in the context of the mechanism of action of the venom and melatonin, is that there are many receptors, cellular receptors in the cell surface that are called receptor tyrosine kinases, abbreviated here as RTKs. One of these receptors is HER2, but there are many others, which are very emerging targets for cancer biology in general. And they're very important in that they activate growth signaling pathways from the cell membrane, and they lead to tumor growth, tumor spread, and in many cases, resistance to therapy. And in the case of triple negative breast cancer, so there is one receptor called EGFR, epidermal growth factor receptor, which is overexpressed in about half, so approximately 50% of triple negative breast cancers. And this receptor is also pretty overexpressed, so overrepresented in approximately 60% of all the ovarian cancer. So those are emerging anti-cancer, targets for these tumors. And so to summarize this, we're looking at novel therapies for triple-negative breast cancers and ovarian cancers, which don't have any targeted therapy available. These tumors are typically treated clinically with invasive procedures such as surgery, also multimodal chemotherapy and radiation, which are very toxic to the patients. And what we're looking at in the lab is novel forms of treatment, and particularly one of the problems of these tumors is that they develop drug resistance. So this is particularly important during metastasis, and that's one of the problems why drugs directed against RKT, so receptor tyrosine kinases, are not that effective in the context of these tumors. So honeybee venom is being, so I'm going to do a bit of a switch on introduction on bee venom and melatonin. So bee venom has been shown to be an incredible source of natural therapeutics, so natural products that are either antiviral, they're also anti-inflammatories, so they're used to treat inflammation, antibacterial, but it's also been shown that bee venom is also a source of anti-tumor molecules for many different tumor types, and there is plenty of evidence in the literature that bee venom could be used to treat many different malignancies, but particularly breast cancer. However, what we are going to do here today is to assess what are indeed the components of the bee venom that can be harnessed to treat a specific subtypes of cancer. So in other words, while the venom was known to have anti-cancer properties, we still didn't know what can be the underlying mechanism by which the venom and the molecular components of the venom specifically kill the cancer cells, and that was unknown. And so today I'm gonna hopefully give you some insights as to how bee venom, what are the molecular determinants of specificity of bee venom to kill these breast cancer subtypes. And so one of the main components of the venom is melatonin. So venom is composed of many different substances. The predominant component of the venom is a small peptide named melatonin, which represents about 50% of the dry weight of the venom. And that is not the only component. There are also a variety of other peptides, enzymes, and amino acids, and possibly smaller molecules that are important possibly important in keeping up with the anti-cancer properties and the properties of the venom. So the main component, as I mentioned, is melatonin. And what is it, melatonin? So melatonin is a small peptide. So it's shown here. It's 26 amino acid long, so relatively small peptide, and it folds as an alpha helix. And what's important is that melatonin, the cytotoxic properties of melatonin in killing cancer cells, they're related to the capacity of melatonin to form these spores on the surface of the cancer cells. And they're about approximately less than 8 nanometers big. And what they do is that you have an association of all these melatonin peptides that form a pore, and that facilitates the entrance of small molecules inside the cell. So melatonin possibly is changing the permeability of the cell membranes, facilitating the entrance of biologicals inside the cells. But the mechanism by which melatonin will disrupt the growth of cancer cells were not well known, particularly in breast cancer. So what I'm going to go today is go over the works done on exploring the capacity of the bee venom to kill specific subtypes of breast cancers that are aggressive, such as the triple negative and the HER2. We're gonna look at the mechanism by which Venom kills breast cancer cells. And then an interesting aspect therapeutically is how we can evolve melatonin so that can more selectively target cancer cells, which is very important if we want to evolve melatonin as a therapeutic for breast cancer. And finally, whether melatonin can be combined with chemotherapies or other emerging therapeutics to increase that anti-cancer effect. So the work that we started on Venom really started with Ciara Duffy, which was an honors student in my lab in 5 years ago. And Ciara started her honors really looking at the capacity, exploring the capacity of the venom to kill different subtypes of breast cancer. And we looked at that, and while this is a bit complicated, I'm going to try to explain it in a simplistic way. So here what we looked at is the percent. So what Ciara did is she extracted bee venom from honeybees here at UWA in collaboration with Professor Boris Weyer. And she extracted the venom, she brought the venom in the laboratory at the Harry Perkins, and she actually treated different breast cancer cell lines that we have in the lab with the venoms. And then we assess the percentage of cells that remain alive. So what you see here in percentage is after the treatment of the cells, we're looking at the percentage of cells that are alive. In function of the concentration of the bee venom, so how much venom we treated the cells. And what we have is a panel of cell lines shown here, and we have different color codes indicating which different types of cell lines we have. Now, these cell lines, they are grown in vitro, but they do recapitulate, and we know that, the biology of the different subtypes. So in gray, you have the normal cells shown here. So those are different cell lines that are not tumorigenic. That means they're normal, like they're not going to make tumors, but we can grow them in the lab in tissue culture conditions. Then in purple, those are hormone receptor positive cell lines that are called luminals. And there are the ones that are more benign, so they're considered more benign cell lines. And then in red, we have the triple negative ones, so more aggressive lines. And then in orange, the HER2 enriched cell lines. So again, these ones here are the ones that are more aggressive. So what we saw surprisingly is that the bee venom killed very effectively the aggressive lines shown here, but it didn't kill as much the normal ones. So as a measure of the potency of any therapeutic, really, we often refer to this indicator that's called IC50, which is the inhibitor dose 50, which is the concentration of the agent which kills 50% of the cells. And as you can see, a good therapeutic is the one that's going to give you a good distance between the IC50 of normal versus tumor. And it was very interesting how the venom was able to very selectively kill better the aggressive lines, which affected much less the normal ones. In the case of melatonin, so melatonin is a small peptide, so we can synthesize the peptide and do a similar study that the venom with this synthetic melatonin, and the profile was similar but not identical. In fact, what we've seen is that the selectivity of melatonin in targeting the more aggressive subtypes was less than the bee venom, so which indicating that bee venom was actually more selective in discriminating tumor versus normal cells. And what will be the anti-cancer mechanism that explains why bee venom, and in less extent melatonin, kills cancer cells specifically, particularly the triple negative subtype, we looked at, first of all, what I want to say is that going back to the mechanism of melatonin, I told you melatonin is a peptide that has this modus operandi, so its mechanism of action is primarily on plasma membrane. And again, making this or promoting the formation of these pores on the surface. And in the first studies, when we look at scanning electron microscopy to see a high resolution, the structure of the cellular membranes after they were treated with honeybee venom and melatonin, we did observe already very much a difference, a morphological difference on the structure of the membranes, again suggesting that melatonin was altering the properties of the membrane, possibly by producing these holes or these pores on the membrane. But we didn't know the mechanism as to why this resulted in such potent and quick cell death. So one of the keys, going back to the biochemistry, was that we went ahead, and one of the hypotheses is like, oh, we test the idea that maybe melatonin and the venom were able to disrupt the binding of these receptors named tyrosine kinase on the surface. This is very important. The binding of the receptors in the surface is incredibly important in activating a signaling pathway, which leads to tumor growth, metastasis, and drug resistance. And we knew this pathway was really important in triple negative in particular. So we tested the hypothesis that perhaps what melatonin was doing in the surface is altering the activation of the receptors on the cell surface, which then inhibited that signal inside the cell, which led to growth and proliferation. And we did indeed test this hypothesis by using a technique that's called Western blotting, in which we measure the degree of phosphorylation of the receptors on the membrane. And indeed, what we discover is that actually both melatonin and honeybee venom were able to inhibit very quickly, in the order of minutes, the activation of the receptors on the surface, which can be measured by that phosphorylation. Consequently, what it did was then inhibit the intracellular signaling, which we can monitor by measuring two more phosphorylations on protein named AKT, which is very important protein in breast cancer. So again, we look at AKT, it was, the hypothesis indeed was correct. So the idea is that melatonin and venom will both might be inhibiting that important signaling pathway, explaining why perhaps these tumor cells died so quickly in the order of minutes. So the mechanism, obviously this is a simplified version of the mechanism, but the next question that we address is whether we could obviously engineer melatonin so we could make it more selective for cancer cells. And that goes back with the fact that venom seem to retain more a selectivity profile, discriminating better between normal cells and tumor cells. So what we wanted to do is go to melatonin, to the peptide, and see, can we then re-engineer melatonin so we gain selectivity? So what we did was actually take advantage of what we call targeted peptides. And targeted peptides, including RGD containing sequences, are peptides that can home melatonin into particular membranes. And in the case of the RGD containing peptides, so this is a small sequence here called an RGD sequence. We call it RGD1. So it can be fused to melatonin to provide an address, a tissue address. So right now, this melatonin is now going to be preferentially binding cancer cells, which overexpress the receptor for these small peptide. Now, we knew the receptor for RGD1 for, in general, RGD-containing peptides, their molecules named integrins, which are very much overexpressed in triple-negative breast cancer cells. And our lab has done, we shows RGD1, particularly this is a small peptide, because we have shown in the past that when this RGD1 is linked to other peptides, named inhibitory peptides, their target more, the resulting peptide fusion target more selectively these triple-negative breast cancer cells. And we have published this before. So what we wanted to do is give help the targeting of melatonin by adding this guiding sequence, which will help to target melatonin more effectively into the triple negative, so hopefully discriminating better between tumor and normal. And what we have found is that indeed the fusion of RGD1 melatonin, and this is a peptide that's now we have intellectual property, and the idea is that this peptide could be potentially targeted this triple negative without giving toxicity or bystander effect when is injected in animal models or in patients. So what we have found it was that the RGD melatonin fusion, what we call the engineered fusion, was able to target the surface of the triple negative breast cancer cells, as you can see here, with an anti-melatonin antibody that we developed in the lab. So in contrast, the untargeted melatonin, so the normal melatonin, actually it is present not only in the plasma membrane, but is also present inside the cells, but in less extent in the plasma membrane. And here, as a comparison, you see the Venom. So it sounds like the targeted melatonin was more distributed or targeted in the membrane of the breast cancer cells, which overexpress the receptors for the RGD1 peptide. And this resulted indeed in a more selective killing profile when we look at the capacity of RGD1 to melatonin to kill. In this killing plot, you can see the melatonin is a lot more efficient in killing tumors than normals, than the untargeted melatonin. Again, we're looking at the IC50. And so again, this is the distance that you want. A good therapeutic will have a selectivity. And so the targeted melatonin had more selectivity than the untargeted melatonin, and this is what we wanted. In ovarian cancer cells, the results are similar. Ovarian cancer cells also overexpress a variety of integrins that are target of RGD1 peptide, and this is work has been done, is unpublished by Edina Wang in my lab, and we have several ovarian cancer cell lines, and in here what we have is normal melatonin with a relatively high IC50 here, and this is a mutated form of melatonin which do not have any killing effect. Now, this is the untargeted melatonin, so the normal melatonin, the wild type. When we engineer the RGD melatonin and we treat these ovarian cancer cells, what we see is a much better efficiency. Approximately, it will be about threefold, a bit less than threefold, but we actually seen an improve on the activity with the targeted melatonin in ovarian cancer. Promising reagent, we hope now to continue some more work with this targeted form of melatonin, which is more selective. And now the next question is that was to ask, well, can we, I told you before, one of the problems of both triple negative and high grade ovarian cancer is that they tend to develop resistance to chemotherapeutics. And so one of the question was, can we use melatonin to actually improve the response of these tumors to chemotherapy, which is a really big problem in patients. And so what we tried, first of all, in breast cancer, we looked at, again, these studies are first in vitro. So what I'm showing here is an example of a triple negative breast cancer cell line, the T11 line. And these cells were treated either with chemotherapy, so this is docetaxel, with Venom, or with a combination of both. And what you see here is that the combination actually improved the killing of these cells when you compare over single treatments. So and that happened in less extent with melatonin as well. This is the untargeted melatonin. So in conclusion, this is what we call a pharmacological synergy. What it means is that when the two components are added together, the chemotherapy and the melatonin or the Venom, the effect is stronger than just the simple addition of the effect of the two treatments when they're given alone. So you have a synergy. And that's very, very important. As I mentioned, a lot of these triple negatives, they develop poor response to the chemotherapies. This is an example in ovarian cancer. Ovarian cancers are treated usually with platinum compounds, for example, cisplatin. But again, the problem is the resistance that the patients acquire. So what we found very interestingly here as well is that melatonin, again, it was a synergistic benefit in having the combination of melatonin with platinum. So these cells are quite resistant with cisplatin to begin with, but the addition of, in this case, is the RGD melatonin. It's helping the cancer cells to be eradicated by the combination. So very encouraging data, given the fact that we believe this could be very important in ovarian cancer, that, again, these patients develop resistance to platinum. And we hope it could be a potentially very important combination treatment. So then what happened in vivo? Well, we done some work in animal models. Obviously, we haven't entered clinical trials. What we have done is what we call preclinical trials. And in this case, what we do is we implant the triple negative breast cancer cells into what we call subcutaneous tumors. And these animals develop tumors. And then when the tumors have a certain size, usually they're palpable, so they have less than 1 centimeter. They have about 100 cubical millimeters. Then we start treating the tumor. And what we did is we treated with melatonin. In this case, the treatment was untargeted melatonin. Here we had a chemotherapy named docetaxel, and then a group of animals was co-treated with melatonin and docetaxel. And again, the data in vitro, it consolidates the previous data we had in cell line, showing that it was a synergism. So what you see here, that the tumors were even regress and maintain very, very small. In fact, during the course of the treatment, when they were combined with melatonin. We could see the tumor by technique named also bioluminescent imaging. And again, what you can see here is the tumors that were treated with a combination of melatonin and chemotherapy were a lot smaller than the ones that underwent single treatments. So again, a very encouraging data in preclinical studies. So we hope to bring this knowledge to further with other models. But I just, this is one of my last slides. I just want to show you that indeed we looked at also some of the markers that are involved in what we call in the anti-cancer immune response. This marker here named PD-L1 is a very important target now for what we call immunotherapies. And we believe that, so that's another line of research in the lab. We're looking at whether melatonin in particular could enhance the anti-cancer immune response, and that's something we're working at present. So what happened is that we've seen a decrease on the PD-L1 levels in tumors that we're treating with docetaxel. This target is involved in immunoevasion, and it reduces the efficacy by which the T cells might eradicate the tumor. So the fact that this marker is down in the treated tumors, makes an exciting new line of research for us, because the idea is to harness melatonin to empower the underlying immune response to be able to eradicate the tumor, so empower the immune system to eradicate the breast cancers, and that's something we're exploring. My last slide is unpublished data, and again, I told you that there is a synergy between that we observe many lines of evidence that the treatment of breast cancer cells, both in vivo and in vitro, leads to an increase in efficacy of chemotherapies. And the principle behind this finding is possibly the permeabilization of the cell membrane. So I told you that melatonin forms these pores, which might facilitate the entrance of small molecules and chemotherapies inside the cell. But new data in the lab show that this can be the case too for not only small molecules of chemotherapies, but also what we call mini proteins, which are many of them have tumor suppressive capacity. So there are many molecules that have a tumor suppressor capacity, but they're not able autonomously to enter the cancer cell. So what we have done is make combination treatments with melatonin, and in vivo we have shown that we obtain really very potent results in eradicating the tumor in vivo by looking at combinations of melatonin with small proteins that have tumor suppressive function. And I cannot tell you the name of the protein here because the stuff is recorded and is under intellectual property. But this is really opens the door to use melatonin to transport larger cargoes that we initially thought. So just my last slide was model of action and of melatonin. So again, going back to this is a sort of a conclusion slide explaining what we think is the mechanism of action in melatonin in killing specifically these tumor cells. So what's the specificity? We believe that melatonin, it gets integrated in the plasma membrane, making these little pores, which facilitate the entrance of therapeutics in the tumor. But by doing so, we believe that melatonin is then disrupting the properties of a cellular membrane, and is disrupting the interaction that occurs between receptor tyrosine kinases. So these receptors have to be close together, in close proximity, in order to get activated. And we spoke about a measure of the activation of the receptor is this phosphorylation here, which occurs after the two receptors have found one another. So by interfering with the capacity of the receptors to form what we call homoheterodimers, we are actually inhibiting the signaling, which is really important in maintaining the cell growth of these cancer cells. In another hand, the presence of these pores might facilitate the entrance of other molecules, which might actually harness other signaling pathways other than the one that melatonin is touching. So what you have is a mechanistic synergy between the two reagents. And the fact that we can choose not only melatonin, we can obviously engineer melatonin to make it more selective so that it can recognize this membrane specifically. We also have a repertoire or an armamentarium of molecules which can be used in combination with melatonin to hopefully devise more specific and effective, what we call personalized therapies for these patients, hopefully preventing the risk of, these tumors coming back or to become resistant. So the idea is to develop personalized treatments for these patients. So conclusions that we found that those are the take-home message. Take-home messages are that Venom turns out to be more specific than what we call the untargeted or normal melatonin. Actually, that was surprising that the Venom was more selective and make us to think why. So we are now collaborating with Julia Graf and Liz Barber and with UWA to actually find out, the Department of Biochemistry actually found out what are the other components of the Venom which might help melatonin to work better. And we are actually working on that line of experiments. Hopefully identify molecules which might help melatonin to be more specific. So that's one angle. The other message was that deals with the mechanism. So we found out for the first time that melatonin targets the plasma membrane and dysregulate cell signaling, possibly by interfering with the interaction of, by the activation of these growth factor receptors on the surface. Now you're going to ask me, well, I mean, if Venom is better than melatonin, then what you're going to do? Well, the response to that is that there is ways to target melatonin and to make it more specific. So one way is to use these targeted motifs such as the RGD, but there are other motifs that we can employ to make these peptides more specific for the cancer types. Eventually do personalized medicine with these peptides. Lastly, what we found is that melatonin mediates the internalization not only of the small molecules, but also cargoes that are traditionally considered difficult to internalize, such as the small proteins, which are or what we call mini proteins, and those have tumor suppression activity. So the discovery that melatonin can work together with these proteins holds high promise in the future. And another line of interest that we have in the lab obviously deals with immunotherapies, because both breast and ovarian cancers they do not respond to immunotherapies. Most breast cancers don't respond. So the idea is to that we're exploring is harness melatonin to empower the immune system to attack the tumor, and that's another line of research. And I just want to thank my lab, and the collaborators helping us to obviously to get this research out and about. I want to thank Ciara Duffy in particular. So that's Ciara here. Ciara started her honors in this project, so then finished the PhD. And Annabelle Sorrell and Eddie Wang, they were really, helping into the animal models. So they were able to test all these combinations in vivo, and they're being really important into helping Ciara to test the melatonin in vivo in preclinicals. Emily Golden and Eleanor Woodward. Eleanor, not only she has helped me do this talk, because it turns out I had a broken finger, and she actually made the presentation for me, so thank you, Eleanor. But she's been critical, really, she doing all the functional studies, really, and the microscopy. So Eleanor was very important into looking at where is the melatonin and where does it go in the cells when we target it. Emily Golden here. So that's Eleanor, and this is Emily Golden, and Emily did all the molecular modeling of melatonin, and there is a lot of work that I haven't talked today because of the time, but into engineering melatonin. So there is places in the melatonin that we can still have room for engineering, and this is what we're trying to do make melatonin more effective. So then the collaborators were Boris at the Cyber Center for the Beer Research here at UWA. He was, the other supervisor of Ciara, who provide all the Venom expertise. Then I want to really thank Julia Graz and his Liz Barbara, and Daniel Juarez Payes, who is a master student and that's been conducting Venom research at the moment. And Danielle is looking at molecules in the venom that can help melatonin to be more selective for cancer cells, and all our funding. I also want to thank Andy Redfern, our oncology, Kevin Flecher, and Elizabeth Johnstone, who did all the receptor experiment for us. And I'm sorry this talk is a bit long. I hope it's not a problem, and I'm looking forward to seeing you in the questions and answers. Oh, thank you very much.